Great. So it is my great pleasure to introduce Professor Archana Anand, an assistant professor of biology at San Francisco State University. Archana got her bachelor's of engineering at the National University of Singapore in 2007, and also an MBA, but I wasn't sure if that was also in Singapore.
Yes.
Yes. Okay. Really amazing. 4 degrees. Dr. Anand has 4 degrees. I am so impressed. Went on to get a master's of science, and looks like you shifted direction towards ecology and did a PhD in ecology and biodiversity, much like Dr. Seth Raman, I think. Saw the light, right? And then postdocs at both Stanford and Columbia, and has worked ever since in epidemiology and virology. It was wonderful. Lots of awards and honors, and 4 just in the last year, I noticed, including Dr. Anand is now a health equity scholar. 2023 has published a lot of really interesting papers in tough journals like Microbiome and Science of the Total Environment, and water research. This is really exciting stuff. So I'm really excited to hear what she has to tell us about COVID. So without further ado, please.
Thank you so much, Scott. That was a fantastic introduction. I'm so humbled, and you're very, very kind. Thank you. Hi, everyone. Good afternoon. I'm glad to be here with you. I know how the end of the semester is crazy. I feel you. I feel just the same. And I really appreciate you all taking the time to be here to listen to me. I'm now going to share my screen. I'll see if this works. Can you all see the fishes? Oh, great. Okay. So before I begin, I want to acknowledge all my mentors, my colleagues in my previous institutions, funding agencies, and collaborators without whom this work, all the work that I'm presenting would not be possible. This is what I'd like to take you through today. I want to, Scott already mentioned this, I want to take you through my journey as I migrated from engineer to biologist. And I want to spend some time talking about how I got into the field of wastewater and began to love wastewater. Wastewater is the new gold, and I'm going to tell you why. And I'll also spend a lot of time talking about SARS-CoV-2 tracking, and hopefully you'll catch the different methodologies that I have used in all these different fields, and that will connect to this, the purpose of this seminar series, which is focused on bioinformatics. This is Marina Beach. This is where I was born and raised. I'm from Madras in South India. I grew up in a family of teachers. Both my mother and my grandmother are teachers. So I guess it makes sense that I thought I should do this as a profession. Or I wasn't sure if I was left with any other choice. It was the only thing I saw and was very inspiring through my childhood. As Scott mentioned, I did graduate with an engineering degree in Singapore, and I worked for a couple of years. I even got an MBA because I thought management would be a good way to kind of mix with the engineering degree. But I wasn't particularly satisfied with what I was doing. I was working in Shell Petroleum as a reliability engineer. I would climb boilers and inspect. And then I took on another job in sustainable packaging. And when the environment focus came into that job, I thought it appealed more. So I traveled a little bit. I took a break for a year. I visited Africa, the Himalayas, places that I didn't take time to see. And this was also when I met my partner, who's my husband right now. And I decided to enroll in a master's in environmental science. And I did this in the University of Hong Kong. This was also where I met my PhD advisor. That's Dr. David Baker on the floor in this picture that you see. And David at that time was working on some really interesting projects looking at human impacts on marine ecosystems. So I really wanted to do that kind of work. And when I approached him, David said, okay, you're an engineer, I'm a biologist, so let's find middle ground in chemistry. And so it began. He had just received funding to look at nitrogen transformations within wastewater. And he wanted a person who had quantitative skills, but also with an interest to apply it in biology. So I was a really good fit for just that project. But before I talk about this a little more, I want to tell you why all this matters. Just 10 rivers carry over 90% of all the global marine litter into the oceans, and 8 of these rivers are in Asia. Everyone is talking about plastic. We've banned the use of plastic straws. This is great. But what about sewage? Do you know where your sewage goes? Do you know what happens after you flush when you use the toilet? Most people do not know the answer to this question. And over 90% of all the world's wastewater goes to the oceans and lakes, wherever they get discharged, completely untreated. Now, wastewater is rich in nutrients, like nitrogen and phosphorus. Nutrients are great, but when in excess, they exacerbate the growth of some microbes over others. And this often aids the growth of some microbes, and they deplete resources that are meant to be equitably shared among all organisms in that particular habitat. So it results in an ocean that's out of balance and results in a condition called eutrophication or dead zones that you may have heard in the news. Now, this was interesting to me because in Hong Kong, they dump over 1,000 Olympic-sized swimming pools equivalent of treated sewage. So I'm going to repeat that: 1,000 Olympic-sized swimming pools equivalent of treated sewage into the ocean every day. But despite this, Hong Kong has way more hard coral species than you can find than in the Caribbean. So I was curious to see what was going on in the marine environment. How are these surviving? Are we having Godzilla corals? Or is there something else special about the ecosystem that is interesting for us to see and could be extrapolated to other megacities like Los Angeles, Singapore, several other parts of the world? So, and that's how the whole process of my PhD and the research story began. So I had the glamorous task of sampling wastewater across 18 sewage treatment facilities. And I was really, really excited about this. Nobody wanted to do this work. And I didn't have an assistant or any of that anything of that sort for a while. So it was just me doing this work. And it was fascinating because the government of Hong Kong was extremely supportive. They gave us access to these facilities, and they also shared all the data that they had collected on the marine environment. So there were several good things that came out of this effort. But I didn't want to focus on just this in this talk. So I wanted to talk about this because it should give you an introduction as to how I got into the whole realm of wastewater. At that time, there were many research groups that were looking at charismatic organisms. They were looking at the Chinese white dolphin. There was the finless porpoise, turtles, cuttlefish. But over 70% of life in the ocean is either unknown, unnamed, or invisible to the naked eye. So I hope you can see where I'm moving you to. I'm bringing you to microbiology and the whole point of bioinformatics for this talk. And that's how I got interested in microbes. But to do this, I had to learn an important skill. I had to learn to scuba dive because my interest was to look at human impacts on microbes in the receiving environment. So I got certified in open water, advanced open water, rescue diving, and scientific diving. And then I began to use several methods to quantify this. So in the next slide, what I want to show you is an outcome of an effort where I used 18S amplicon sequencing and CO1 meta barcoding. I'll speak a little bit about that. But before that, to orient you, this is where we are. This is Asia. I know it just looks like a big chunk of blue and green. And this is Hong Kong. I'm sorry if it's unclear. I'll do my best to orient you. So this is Hong Kong's special administrative region. The water body around that mass of land is the South China Sea. The gradient that you see in the water body, which is slightly brownish, is owing to the Pearl River flushing fresh water into the South China Sea over here. So this is almost like a natural laboratory setting, because as you move from the west to the east, the salinity gradient changes. So it moves from more brackish, like freshwater conditions, towards it being more marine. And we see something similar for pollution as well. It's much more polluted on this side. When I say pollution, I mean nutrient concentrations that I mentioned, higher nitrogen concentrations in the west, and it decreases as you move towards the east. Now, the heat map that you see here shows you 7 sites, 1, 2, 3, 4, 5, 6, 7. These sites you can see using the same colors. I guess not, yeah, sort of the same colors. The red is high impact. So you see these 3 sites highlighted in red are highly polluted sites, compared to the blue sites highlighted in this map that are relatively low in pollution. And we also have a medium site, a site of medium impact that could not be put in either of these groups because it was more flushed with the fresh water as well as the marine waters. So that's why it's a medium site. And now I'm going to present what, before I do that, what we did was we deployed these mini ocean hotels. They're just structures that are PVC plastic plates, about 9 plates. I leave them in the seafloor for a year or 2, after which I collected and looked at everything that had colonized the plates using the CO1 barcoding as well as photography. Now, what was fascinating was that the green line here is the South China Sea, where I had all my samples. And you can compare them because it's a standardized method. We compared them with the Red Sea, Florida, Virginia, and Singapore, all of which are not as polluted as the sites that I was studying. And what was shocking was the richness that we saw in the sites that were polluted. Well, this includes all the sites, right? That includes both the high impact and the low impact. But you also see in this figure, I show you high impact, which is in the purple line, and then the low impact you can see with the orange line. And they are both higher than what you can find in, say, Virginia, Florida, or Singapore. And of course, the system I'm talking to you about is a temperate system, not a tropical system like Singapore, which is all the more interesting because a tropical system is likely to have way more biodiversity than a temperate system should. So this was interesting. Now, what we also observed was that what we also observed was the site that was the medium impact site had the highest biodiversity compared to the sites of high or low impact. And we're still working on figuring this data out, but I wanted to share this because it is aligned with something called an intermediate disturbance hypothesis. So if you want to take time off your already existing packed schedules when you're on the BART or on the train, you can look up this hypothesis, which says that a site that is characterized by not too high disturbance or not too low disturbance, or perhaps intermediate disturbance, is what is likely to contribute to higher biodiversity. And that's what we saw in Hong Kong as well. So now this was also when I had my first child. This is Advik. He is now 7 years old. And I continued this work. What I did was I also began to look at sediment near the structures that I deployed in the seafloor. The video, of course, is not showing me collect sediment, but what it's showing me do is deploy tea bags. That's what you see here. So what I also did was bury tea bags, which are just green tea and red tea bags, rooibos tea bags that you drink. Red tea, rooibos tea is very, very tasty. I like it a lot. So we deploy this to get estimates of carbon storage and rates of decomposition. And the idea was to see if we could attribute these to certain functions that these microbial communities are performing in the seafloor. The paper that I highlight here is using 16S, which is another marker. So I did say that I will speak a little bit about it, but I guess I skipped that. CO1 and 18S, I'm sure you're all familiar, but if not, then they are different markers that are used to, and they are universally conserved. 18S and CO1 are conserved in eukaryotic species, while 16S is often very well conserved in bacteria and archaea. So they're used to look for different groups of populations. And in this case, I used 16S amplicon sequencing. I also used metagenomics. And of course, none of these works were just done exclusively by me. I collaborated with my PhD classmates as well as my postdocs in the lab and neighboring labs, several of whom are in the papers that came out. So I'm going to now share you just one figure, and I hope it's not too busy. I'm going to take some time to walk you through what you see here. On the left is data from the 16S amplicon sequencing of the sediments. And here I'm showing you 4 sites. So the 4 sites are highlighted here. All I want you to think about is that as you move from the red to the orange, it's highly polluted, and it gets lesser and lesser polluted as you move down to the orange, which means CI is the most polluted site and TPC is the relatively least polluted site. It's definitely not pristine. There's no such thing as a pristine site in Hong Kong. So we would just call it relatively less polluted. This figure shows you a group of microbes called the sulfate-reducing microbes. These microbes have been associated with carbon cycling and are known to be somewhat connected with it. There are several studies that have looked at them, but exactly what they do is still, we're still working on it. It's still fragmented, and it's not as well understood. So the first thing that this figure should give you, or at least tell you, is you see lots of microbes of this group in the sites that are more polluted. And as you get to the site that is not as much polluted, they decrease in abundance. So the y-axis gives you relative abundance, and that's a clear shift in these groups of microbes that we see as we move from the most polluted site to the least polluted site. So there was something going on. Now, I wanted to pair this data with what I was seeing for the ecosystem function assays. And what I mean by that is the teabag method, which does not give you any microbial data whatsoever, but all it does is helps you quantify how much the microbes have degraded organic matter and how much they have left behind after the degradation that could help you figure out how much carbon storage capacity is available at that site. So on the x-axis is organic matter stabilization, and that just means carbon storage. And on the y-axis, you see decomposition rate, which is the rate at which microbes are degrading organic matter. Now, again, you'll see the same colors. There's red for CI, which is the most polluted, and TPC, which is the least polluted. So if you see first, I'm only focusing on the colored groups here. So what you see is the CI has a relatively higher rate of decomposition when compared to TPC, which is also our more cleaner site and could have a lower rate of decomposition. So the sites that had more of these microbes were also degrading organic matter faster than the sites that had lesser of these microbes that were doing it relatively slower. Another thing that we found was that when you look at this in just the S scale, the more polluted site was doing very poorly when compared to, for its carbon storage capacity, when compared to the cleaner site, which did much better. Now I'm going to have you move your eyes to the black dots that you see here. And these are reference points, because this teabag method that I'm saying is not something that I came up with. It came up in 2013, and the authors have since used the method to quantify these two very parameters in several habitats and geographies. And they have this data that they have publicized. So we overlaid our data with their data to see how our eutrophic or polluted marine environment compared to what they had. And what we did find was something that was very polluted did do poorly, but something that was not as polluted did not do as badly. So this was sort of a story of hope maybe you can call it ocean optimism, that despite several environmental stresses, there are communities that are able to perform functions that, at least to a certain extent, that is required or could have been possible without these several stresses put on them. So I hope everybody's with me so far. This was also when I moved to the Bay Area for the postdoc that Scott mentioned at Stanford. I'll speak a little bit about that shortly. But when I came, I was actually looking at sandy beaches. I was not doing any work on wastewater or SARS-CoV-2. I started off doing that. And this was also when I had my second child. This is Adit, and he's a pandemic baby, which is why he has a onesie that says, "My parents did not practice social distancing." Now I'm going to talk about just sewage, which is what I'm really, really excited about. And of course, sewage was the baseline of everything else that I shared too. So sewage is becoming a gold mine, literally as well as figuratively, literally because gold is being mined even in sewage in several parts of the world, more so in countries like India, where gold is being found in wastewater in relatively larger amounts. Not just gold, also silver, metals that are used in consumption of food and sweets that are being served exclusively during huge festivals. And this is something that is being explored as a possibility. So just in case you didn't know, something to pick your brain about, or maybe you'll look this up shortly after this call. But today I'm going to speak about wastewater-based epidemiology. So before I continue, I have a question. Does anybody recognize who this person is? You could feel free to type it in the chat if or unmute yourself. Oh, great. I see Scott saying yes. So this is Jon Snow, not from the Game of Thrones. Dr. Snow was a British physician, and he has played a significant role among several other scientists, of course, but in this context for wastewater-based epidemiology. And this was because there was a huge cholera outbreak that emerged in Soho in London. And at that time, cholera was thought to be airborne. And when Dr. Snow proposed that it could be transmitted perhaps through another medium, in this case, water, his theory was trashed and was not accepted at all. But what Dr. Snow did was he came up with this map that you see here of different pumps, and he narrowed down one particular pump that could have been bringing contaminated water from the River Thames into the city that people were using to consume, as well as maybe wash dishes and use for several other domestic purposes. And when he found that it was indeed the cause for the spread of cholera within a certain group of the population, and of course, at that time, this was not accepted, but later it was understood that the study of diseases in populations was, he essentially founded that, or contributed to that significantly. So here now I want to talk about how I came into this concept as well. I was here, I just moved to the Bay Area in 2020, June of 2020. This was when I just obtained a postdoc position at Stanford. The group was mainly looking at microbes in marine ecosystems and habitats, very similar to what I was doing. But they also were collaborating closely with a group that was doing wastewater work, and they were looking urgently for scientists who had experience handling wastewater. So I think I was just at the right place at the right time. I was lucky. So I got sucked into these incredible projects. And if there are students here, I would encourage you to network as much as possible and share your interests with as many researchers as possible. You never know who would want you at what time in your life. So that's what happened to me. And I didn't expect this. So at first, what the group was interested in doing, which is the group that I became a part of, this was in the BAME lab in Stanford. We knew that, in fact, we did not know. So we thought that the virus, SARS-CoV-2, which causes COVID-19, could be shed in feces of individuals that contract the disease, and we are likely to see it in wastewater. But we had no idea if we can correlate SARS-CoV-2 concentrations in the wastewater to the number of cases we were seeing in hospitals. Now, I'm going to take a step back and tell you that this is not new. This is not something that just came up in 2020. This has been around for a very, very long time, for several decades. And in fact, wastewater-based epidemiology, essentially tracking diseases and the spread of diseases in wastewater, helped India stop a polio outbreak. Recently, the virus could be seen in the wastewater up to 3 weeks before any symptoms began to appear in an individual. I'm sure you would have heard in the news about the virus being tracked in wastewater in the United States, in New York as well, in September 2022. So there's a lot of interest in using wastewater epidemiology to track several other pathogens. But now I'm going to speak just about SARS-CoV-2. Now, people were falling sick, and there were hospitals that were getting full. And there were researchers that were were looking at the virus themselves. There was an urgency to create a vaccine because this was in 2020 June, and no one knew what we were dealing with. There were just populations getting sicker and sicker. And Google was doing its job of tracking people buying ibuprofen and lozenges in, say, pharmacies and CVS, Walgreens, and to use that as a measure of how much the disease has spread in populations. But some of them, people were getting tested. Not all of them were getting tested, but everybody uses the restroom. So we knew that wastewater would be a great way for us to truly have an anonymous, because we can't track this back to anybody, ethical as well, picture of the spread in a population. So this study here is the data I'm showing you. I know it's a very busy figure. What I'm hoping you will take from it is these are 7 sewage treatment facilities, huge treatment plants in all over the United States. And they serve totally about 2 million people within the United States. And the idea was for us to just see if we can correlate the clinical cases that we're seeing to the gene copies of N1 and N2 of SARS-CoV-2. So now I'm going to stop there and tell you what N1 and N2 are. I have a figure about with the genome of the SARS-CoV-2 virus itself, but essentially at that time, the copies of the gene N1 and N2 were considered to be the most reliable for us to track the abundance of the virus in the wastewater. So that was the only reason for choosing N1 and N2. There were other proteins that could have been used, but then the methodology development happened concurrently among several research groups that shared it with each other. And N1, N2 took off. There was ease of development, and hence the reason to use this method. Now, what you can see here is all the graphs on top show you the new cases of COVID-19 between March 2020 and July 2020. So if you see here, this is March, and of course it goes on to September over here, 2020 at the bottom. And all the figures at the bottom, so these dots, the black and red dots, show you N1 and N2 gene copies. Now, if I were to ask you to just not focus on anything in particular, but just look at the slide, step back and look at the slide, do you think the trend of the N1 and N2 abundance matches the trend that you see in the clinical cases, at least roughly the highs and the lows? Sort of, maybe. Yes, so we did see a correlation. And we did see, indeed, yes, we did see a bit of a delay as well. So first thing we concluded was, yes, it's correlated. But second, could there be a lag in some? We don't know which one was lagging. Was it the wastewater or was it the clinical cases? Now, that was something that our group did not work on. There was a neighboring group that looked at whether we could use wastewater to track the disease before symptoms begin to appear. And the answer to that was yes. It was another group also at Stanford, but remember at this point, it was not just one university. There were several universities doing amazing work to figure this out. So I was just part of one research group that shed light on this, but there were several other researchers working on similar topics, and we all arrived at this collectively. And the research group that I'm highlighting here did find that you could see the virus in the wastewater up to 9 to 14 days before an individual begins to exhibit symptoms. So then we began talking to counties. And once we saw a rise in the wastewater, we would talk to them and say, look, we're seeing a rise, and you are likely to find individuals exhibiting symptoms perhaps in the next week or two. So you could account for hospital beds or ICU accommodations could be made to make sure that populations are being cared for in the right way. So this was really one of the first times where I thought the work that I was doing was really could be translated immediately to the health and care of the public. But the next question that we wanted to ask was, okay, these are like giant treatment plants, but at that time, universities were closed. College schools were not running. People couldn't go into work, and they were all at home. Prisons they had to be at one spot. But then could we perhaps say that this can be translated to a sewer shed of various sizes? Could we do that? Or is the correlation only possible if it's like an extremely large sewer shed of 1 or 2 million people? So this, I just have a video to show you what we did. This is in a dorm at Stanford. And what this gentleman is doing is placing an auto sampler within a manhole. And the auto sampler essentially helps you, it's fitted with a Nalgene bottle. So I'm not sure if you can see this Nalgene bottle here behind this other gentleman's leg. And it has a bottle like that in the auto sampler, and it has a tube that connects to the sewer line. And what it does is it draws, say, 10 to 20 mils of wastewater every hour over a period of 24 hours. So we go back the next day and we've got to pull out the auto sampler and get the bottle out and take it for processing. And I'm just going to give you a minute to think about this. Do you think this would be useful? We could also, of course, go and just grab a wastewater sample. And many researchers have used that too. But maybe can you think about why this would be useful in terms of data? Now, if you want to, yes, exactly. Synchronous batches of data collection smooths out variability in sampling. Exactly. And you're all on the right page here, right track here. Now, if I want to track a person who has COVID-19, who perhaps did not use the restroom at the time shortly after which I'm collecting my grab sample, it's likely that I miss seeing those concentrations in the waste, or those certain his contribution or her contribution or their contribution to the N1, N2 copies in the wastewater that I want to see in the sample, right? But if I were to get a composite sample that is fairly representative of several users of restrooms over a period of time, then I'm likely to be able to catch that. But remember that this is the sampling that we did here is, when I say level 1, level 2, and level 3, they're all sewer sheds, a minimum of 200 people in level 3. So it's still anonymous. We have not been, the idea was never to try to track down an individual, because that is the role of a clinical test. So the idea for us was only to see if we're able to see a trend in the disease and the way it spreads across populations, and whether that could help inform hospitals and other healthcare providers when they interact with these individuals who may have the disease. So what we found was that for levels 2 and 1, which was about 500 individuals and 1,000 individuals, this we could correlate the concentrations very well. But for the level 3, the concentration and the number of clinical cases was a bit hard. It was more easy for us to look for presence or absence. And this is still, since then, of course, people have looked at it and perhaps improved the recovery and looked at better, more efficient ways of using the data. But at that time, the sewer sheds that could be most useful for using this were over 500 individuals at that time. Okay, so now I want to just move to, I hope I'm doing okay on time. I'm coming towards the end, and I thought I'll spend some time talking to you about the other postdoc where I did variant and mutation tracking. So far I was speaking about abundance. And again, coming back to bioinformatics, this is a very targeted analysis. It was using a technique called ddPCR, which is double droplet polymerase chain reaction. And it has since become extremely popular. It is only because of the resolution that you could get of one particular thing that you're looking for, like a needle in a haystack. Now, what I did after I moved out of Stanford, and this was in another group that I joined at Columbia, this is the Chandran Lab, and which was in the environmental and civil engineering department. They have also been experts looking at wastewater, but for very different reasons. They were looking at wastewater treatment and what microbes do in different treatment processes. But their lab was again brought on by the CDC to see if they could perhaps assist in the figuring out how the pandemic was spreading in populations. So what I did there was I looked at two large treatment facilities. One was in New York City and one was in New Jersey. And I looked at how the variants at that time, because when we first started at Stanford, there was no talk of a variant. It was more about abundance and whether it was going up or going down. But by the time 2020 ended, there was, I think, Delta began. And soon after, it was the time for Omicron to shine. And later on, since then, I'm sure you've heard terms that are very confusing and you may not even comprehend, like XYZ.1 or BY.2, which are all recombinant lineages. I can tell you more about exactly what they are if that's of interest to you. So here I'm going to share a little bit about a paper that just came out, I think, about 2 weeks ago from my postdoc effort. But to orient you, this is the genome of SARS-CoV-2. And as you can see, there are several kinds of proteins, as is always the case for several other viruses. But in this case, the big green dot here that that's just come to hover on the genome is meant to show you that most scientists have focused on the S protein, which is called the spike protein. And there's a reason for this, because the spike protein has been traditionally associated with immune response in individuals. It's also the protein that assists the virus in binding with receptors in different types of cells within our body as well. So it makes sense that vaccine development has also focused on this particular protein. But there are several other proteins that we wanted to focus on that could have perhaps contributed to the infectivity and the transmissibility of the virus in populations that we are not seeing because we're so focused on just one protein in the virus. So the idea of this study was to, through this, hopefully, I'll also educate you about how important it is for all the proteins that to be considered, and also to look at things in a holistic way rather than to focus on just one. So if you remember, what I told you was, at first, the interest was only to find who is in the wastewater. Are we seeing SARS-CoV-2 in the wastewater? And then how much of that are we seeing in the wastewater? So that was what was going on. And then there were studies that began to look at, is there something in the wastewater that can help us understand whether there is some mutation that could have a certain functional implication for this virus? Is there something going on, which is why Omicron is behaving in a certain way or Delta is not behaving in a certain way? So to do this, there were several studies, like I mentioned, have looked at the spike, which is made up of the S1 and S2 protein. And again, nothing to belittle that. This is extremely valuable because, like I said, there is a reason why vaccine development is focused on this. But the purpose of the study was to show that there are several others that are likely to be valuable as well. And we also talk about the spike in the study, mentioning them in a holistic way. So again, I have just two figures that I know will appear very busy. So now what I want to show you here is we found a bunch of mutations. Now these are mutations that we found, amino acid mutations, that we found in over 90% of the wastewater samples. So there was something definitely going on with these mutations, and they were persisting in the wastewater wherever we got them from. And we trace these mutations back to the region in the genome that these could be found. And if you notice here, these are the S regions. So if you see out of 13, there are 1, 2, 3, 4 that can be traced back to the spike. But the rest are in other parts of the SARS-CoV-2 genome that are often not studied. Now, on the right side, the heat map shows you where these are found, mostly in different variants. So if you remember, we had an alpha, beta, delta, gamma. There was iota, which was exclusively a New York variant, very special to New York. Lambda, Omicron, BA1 and BA2 at that time. And so the darker greens essentially mean that there was more of that mutation in this variant than, say, in beta. And the light green is just less of that mutation in a particular variant. Now, if you move to the right of the table, and I hope you're all still with me, I show you different functions. That was the whole point of the study, right? To see if the mutation can have a role that it plays for the virus to make it as infectious or transmissible. And yes, so there are several ticks, but there are some mutations for which we could not find a known function because at that time there was not much work being done or it was less understood. So we were seeing some mutations in over 90% of wastewater samples. These were collected from over 2 years, but we had no idea what they were doing in the population. So the paper still, of course, says that we still don't know, and there's work that needs to be done. And I just wanted to hopefully have you appreciate that as well, that there's still so much that needs to be done for us to understand what these mean for populations, not just in New York City and New Jersey, but several of us here as well. Now, this is a figure that shows you the same genome on the left. And what we did was correlate those to death rates, hospitalization rates, number of individuals that tested positive, and number of individuals that were vaccinated fully at that time, and how they could be correlated. So we saw some mutations that were correlated with mortality, heavily correlated with mortality. Which meant that, and of course others as well. So the paper suggests that maybe we should be using mutations as markers of tracking the disease, because if we see a certain mutation increasing, then it would be a red signal, because we would then need to make sure that we take care of individuals, or we account for certain symptoms that we foresee to perhaps worsen. So we can avoid increase in mortality rates or hospitalization rates. We also saw some mutations like the G662S correlated with individuals that were vaccinated. So maybe this was giving them some immunity or resilience to the disease. Again, it's not as well understood. So this is the sort of thing that we discuss in this paper. And I want to leave you with, of course, we want to expand our focus beyond some proteins that we know and are well studied. Could wastewater, of course, serve as the disease tracker beyond SARS-CoV-2? Yes, and that's what I want to end with my lab. I have a few, something to share about what I'm doing right now. And can we use wastewater to go beyond just disease? Could it be used to make connections to nutrition and health, just overall health in a population? So here I run a lab at SF State, and I'm very grateful for the students in my lab. I've only been here for a year. Not everybody is here in the picture, and I've done my best to share their names with you. So the first thing that we're doing is we have changed 4 manhole covers from the 60s on SF State campus and replaced them with holes that have, with manhole covers that have pre-drilled holes. And this is because we're using tampons to sample wastewater. And if you saw that giant auto sampler that was being installed, we're moving away from that because tampons serve very effectively to get composite samples of wastewater that are not grab samples. And it's very safe for the students they wouldn't need to open the manholes and get in and get out of them. So we're working on that. We're also looking at phages in wastewater. That's the next thing that we're hoping to do, and sequence microbes in general, not just SARS-CoV-2, but just who is there to build a baseline of diversity of microbes in the wastewater. And then the other thing I wanted to speak about was we also, this appeals to my other side of me, which is my coastal ecosystem and microbial side, my environmental microbiologist side. So we're looking at oysters in oyster reefs and how microbes in oyster reefs are perhaps contributing to the resilience of these to climate change. So this is my grad student here who's working with some experts from the Smithsonian who have been restoring these oysters already. So we're working to see how best we can support them. And lastly, this is because I have my son goes to school with a mom who works at a vineyard. She operates the vineyard, and they were keen to look at whether soil microbes could contribute to the taste and quality of the wine that they produce, because they want to inoculate wine with a certain native fungal community and not get commercially available yeast to inoculate the wine that they ferment. So these are, again, my students. We've been sampling soil, we've been sampling grapes, not yet the wine, no, but we will hopefully at some point when these grapes get produced and become wine, and we want to see how these microbes contribute to the pungency and the quality of the wine. Now I have a dog. That's the last slide I have. She's the only girl in my life. And she's a Great Dane. But I hope I didn't overdo the time-wise, and I really, really appreciate your patience listening to this. I'm really happy to be here again and can take any questions you have. Thank you. I see some questions in the chat.
Yes.
Scott, should I just perhaps go through those?
Yeah, so they're great. Let me see how I'm scrolling the chat. Here we go. Yeah, there was a, so the first one that I have in the chat is it possible that some genes or some regions could persist? Oh, it got all messed up. Some genes or some regions could persist longer in wastewater than others, or be responsible for them, for the longevity of the virus.
Oh yeah, I see that. Thanks, Scott, and thanks, Mary. Yes, for that wonderful question. That's a great question. I do not know the answer to that, but what we do know now is when this work was being done, we asked the exact question that you were asking as to how long does the virus persist in the wastewater even after you recover from the disease, and the answer at that time was we don't know whether the shedding that we're seeing in the virus is because of new infections, or perhaps people who are infected are going through the symptoms, or they are done with the symptoms and they're still shedding the virus. And so I think there was a study that recently came out that said people can shed the virus for up to 90 days after infection, which makes the whole thing even more confusing. So it is, it could be possible, but I'm at this point unsure. I do not have an answer for that question. It's a great question. Thank you.
Great. Nora is asking, how frequently do you collect wastewater samples, and are there differences between tampon brands for this microbial sampling? God, I love the fact that you use tampon. I love science.
Great question again. How frequently we do this? We do it twice a week on Tuesdays and Thursdays. We've been doing it since July 2023. Right now, we're at a method development stage. I have my grad student Michael, and I have and my undergrad Ben, and several others in my lab who are leading this effort. They are using a method called the nano trap. It essentially uses magnetic beads, which help to separate the microbes, or help us in recovering what we want to look for in the wastewater. And it would be great to replicate something like this in other CSU campuses. But yes, that's what we're doing right now. And we've sent the samples for amplicon sequencing, just 16S for now, to see if we're getting what we want to get. And then we'll go from there in terms of other omics. The next part of that question was, do we have different tampon brands? A great question. We've not explored that just yet. I think it would be a fabulous project to look into. Right now, we're just using, I think, one brand that my undergrad recommended, and I'm just going with his recommendation on this. But it would be something to, worth exploring.
Another one from Laura is, how does wastewater surveillance of COVID-19 take into account that people travel between sewer sheds sometimes?
That's great. Good question too. So yes, this is why we do it in dorms, just so we can make sure that we have a signal that we capture from residents of those dorms. And we know for certain that it's from like a population that is relatively like, around, although of course they could have visitors and things like that, or travelers. But the idea was for us to do it in a campus. If we do it long enough, the idea is we would be able to account for that variability within the same manholes over like a year. So we would still be able to capture a trend. So that's the idea. I'm trying to go over a question.
So, Riley asked, you want to go to the next one? I'll help you.
No, go ahead. Thanks, Scott.
No, that's great. These are great answers. So many good questions. Riley asked, could you track the mutational signatures that have been associated with Paxlovid use and associate those relative rates of purchases or use of Paxlovid, I guess, in respective wastewater catchment locations?
That's a great question. I would imagine yes, as something that could definitely be looked at, and also to track the use of whether we're able to see the drug in the wastewater. And that would be a different line of work. It would not be as much bioinformatics as it would be metabolites, metabolomics. I would think it's an area that's very hot right now. So I think I only know I know a few people working on it, but I know it's extremely interesting to look into, and I would be interested in it too.
Yeah, I guess there's a recent paper from Sanderson et al. We'll have to check that one out. That was pointed out in Nature. Another question from Angus. These are all student questions. It's amazing. Is it possible the environmental conditions in sewage could cause some of the amino acid mutations? That's a wild question.
That's and it could be, you mean like temperature or perhaps who knows? Yes,
are they replicating in this? Is there an idea that they do some replication outside of the body, or not really?
Not really. So wastewater is fairly safe. The virus that you see, the populations are of course you need to take precautions, but then relatively it would be a safer place for you for the virus to be, because they are not infectious at that point. They're dead copies of the virus that we track. But these possible about environmental conditions in sewage, I would say environmental conditions in general could affect the virus in different hosts and therefore translate to observed amino acid mutations in sewage. That's how I would think. And I think that's definitely, yes, the case. We are likely to see different susceptibilities of hosts according to, climate changes. There could be other pathogens that interfere with one pathogen's ability to infect the same host, which could also change. So yes, there could be several factors that you see. Yes.
Avalon has a question, but I don't think I quite understand it. So maybe you can read it. I don't know what prey proteins are, but
were any bait proteins researched in this project, and their respective human prey proteins particularly prevalent in the wastewater? For example, are there more ORFs that target heart cells present in the wastewater compared to other prey proteins? That's a good question. We haven't looked at which particular part of the human body or which cells that these could target. I think the study that I presented is like much more generic compared to this question. I would imagine that this is a very targeted area, and I'm not familiar with ORFs that target heart cells particularly. So it's something, yeah, I'm happy, I can look into it and see, but right now off the top of my head, I'm not sure.
It's a fair answer in science. It's the honest one. And makes study for all you all, right? I had a question for you going back in time. Since I'm doing the Tijuana River estuary stuff with Carrie Sant, and we're doing a lot of stuff. When you were talking back about your system, one thing I noticed, I do some source tracking of water coming out of the Tijuana River and mixing with marine environments. And one of the things you had in the Hong Kong environment, you were talking about modest, and you were comparing it to other parts of the earth, but you didn't talk about the organisms that were actually coming out of the river and mixing. Because if you had a really large river system that was dumping water, fresh water, even in the 16 or the 18S, you're going to get what we saw in our system was lots of things like amoebas and lots of other stuff coming out that I think could definitely really increase your overall alpha and your alpha diversity. Yes. In that maybe if you compared it to Florida, which may not, I don't know if they have these giant river systems coming out, that it might not be so much about the pollution as more about the inputs, or maybe it's a combination of both. What do you think about that? Because I see lots of amoebas and lots of other things coming out and mixing.
That's a great point that you've highlighted, and it's this paper is actually in review, and we did get the same comment from 2 reviewers. I did not review it. And we did respond to that saying, yes, it could be the fresh water that's likely to contribute to this, because it's hard for us to pass out these different variables that could be working towards this. But I also, this might be a bit tangential, but we also did some stable isotope work on the nitrogen transformations to see what was contributing to the nitrogen in the environment. And we did see less of a signal from the Pearl River, but we saw, although this is not part of this paper, it was another piece of work, because this paper was very focused on just microbes. But just since we're talking about freshwater and dynamics, we did observe that well, no, let me back up a little bit. So there's like a giant wastewater treatment plant in Hong Kong, and there's the Pearl River flushing freshwater from mainland China. So there was a political debate about which one was giving a cause to larger pollution. And I was part of a group of scientists that did a great job of putting this whole mess to rest, which was, so I was only a student at that time. And the answer that came out was it was not Pearl River that was contributing to the pollution, but instead it was Hong Kong's very own wastewater treatment facility that was responsible for the nitrogen, inorganic nitrogen in the marine environment. So this could translate to something to do with the microbes as well that we're seeing. But yeah, I'm thinking the Pearl River might actually, the solution to pollution is dilution. And I'm wondering if there's like a lot of fresh water coming in there to make it a little nicer. To your point, also the work that we see in the sites close to the sites that have the Pearl River, yes, in the paper we did respond that yes, we cannot completely eliminate the possibility of the freshwater contributing to the diversity that we're seeing. Exactly, I'm with you on that. Yeah. No, it's really exciting. I want to talk to you more about it. I actually have to go to a meeting. Yes. And I'm the host. But if you do want to reach out to Dr. Anand, you still have questions, I see some in the chat, please feel free to, I assume they can contact you by email and ask. Sure, of course, yes. You're doing such exciting work. Thanks so much for sharing it with us. And is it okay to put this publicly on YouTube? Oh, sure, of course, yeah. Great, okay, well, we will be in touch, because that's great stuff. All right, bye everybody, thank you. Thank you.